So some bacteria can cause disease. They can cause disease by producing toxins, either an exotoxin or an endotoxin. An exotoxin is just a toxin that is secreted by the bacteria. An endotoxin, endo means inside, doesn't get released until the bacteria dies. So they're still toxic, but the bacteria dies, releases the toxin. You can see on the right the classic bullseye rash. That's Lyme's disease. That is carried by ticks, little tiny deer ticks in this area. Bacteria can also be used as biological weapons. And one that most people think of when they think of biological weapons is anthrax. And the reason they think of anthrax is that it can form these endospores. Remember, these little capsules that will protect it for many, many years, in fact. And the most dangerous form of anthrax is the one that's inhaled. So you can volatilize it into the air, people inhale it, and that's the worst way to get it. You can actually survive it better if it goes through your skin, but inhaling it's really dangerous. Prokaryotes can be very useful for us. I don't know if you guys remember the big oil spill out in the Gulf, but prokaryotes will digest up oil and other things and recycle chemicals, including these heavy metals that are from mining and stuff like that. And they can be used to clean up pollution in the environment. So they can be used both in sewage treatment plants and, like I said, oil spills. And on the right, you'll see that the toxic mines, you actually can spray these bacterial mixtures and they will digest away some of the toxins released into the environment. So they're very, very useful when they do what they do best, which is metabolizing. Certainly sewage treatment is a good use of them. Okay, eukaryotes probably evolved about 2 billion years ago from prokaryotic cells. And they're much more complex. They have a defined nucleus, they have organelles, and so probably what happened was you had some prokaryotes that kind of absorbed in other prokaryotes, and then after a while they would start reproducing and you got a full eukaryotic cell. That was about 2 billion years ago. Okay, the nucleus and the endomembrane system probably came from the infoldings of the plasma membrane. So if you think of the plasma membrane on the outside, and if you can think of folding it back and forth, you can get things like your Golgi apparatus, your endoplasmic reticulum, your vacuoles. All that came from infoldings in the plasma membrane that was surrounding the prokaryotic cell. Okay. Mitochondria and chloroplast, I think this is so cool, probably evolved from these aerobic, if it's a mitochondria, or photosynthetic for the chloroplast, what are called endosymbionts, which are just other prokaryotes that came into the cell and were so very, very useful. They're like little aliens living in your cell. They're little bacteria that went into your cell. The mitochondria is very, very good at producing energy. Chloroplasts are very, very good at photosynthesizing, so they were useful. Okay? So here's a picture of probably how that happened. When you get this folding, right, the membrane folding, you can get endoplasmic reticulum, you get your nuclear envelope, you can get your Golgi apparatus from the folding. But look here at this, because I just love this little aerobic heterotrophic prokaryote. That is now your mitochondria. It was a little alien invader that came into a cell and was very useful at producing ATP. All right, here you get the little photosynthetic prokaryote. Well, that is now your chloroplast, and very good at taking energy and making chemicals. Okay. So early scientists to observe cells used the microscope, the lenses obviously, and made sketches of what they saw. You can see here on the right, these are supposed to be nerve cells of the retina of an eye. Over here, these are the nerve cells over here, the retina of an eye drawn, because they didn't have micrographs. They couldn't take a picture, so they just sat down and meticulously recorded what they saw under the microscope. So light microscopes really provided a window to the world of cells. Without it, you really can't see very many cells. When you're talking about things that the unaided eye can see, you're going down to things like as small as a frog egg or something. Very hard to see single cells. So the light microscope really opened up the world of the cell. You can see down to about a micrometer, which is the size of a mitochondria approximately. And it lets us see the overall shape and structure of the cells. It's not super detailed, but it's pretty good. So your typical light microscope will magnify about 1,000 times, so you can see down to about a mitochondria, like I said. And what's nice about them, a couple things. One, you can look at living things. The other microscopes, your specimen has to be dead. But you only get to look at the surface of whatever your specimen is. You can get an idea of organelles in there, but you don't really get to see inside of it. All right, in this picture, you're going to have two different microscopes. On the left, you have a scanning electron microscope, and you can see over here, you get a really, really nice idea of the surface. You can just see the details of the surface. You can't see anything on the inside of the cell, but the surface is very, very clear. And again, these specimens are going to be dead. They can't be moving around when these micrographs are taken. On the right side, you're going to see over here, this is a transmission electron microscope picture and a micrograph, and you can get a much better idea in here of the organelles. So, okay, most cells are microscopic. That's really, we needed to wait for microscopes to be invented before we can see these things. If you look in this range right here, you're going to see most of your plant and animal cells are just beneath what you can see with the unaided eye. You really need the light microscope to see these. Here's your mitochondria. That's kind of the bottom of what you can see right about there with the light microscope. And after that, you need to get into more high-tech microscopes, your electron microscopes, and you'll see the stuff down in this range. The unaided eye does not see anything below not quite 100 micrometers. Okay, the microscopic size of most cells ensures a sufficient surface area. And you say, well, what does that mean? Well, it means that the way nutrients and waste, nutrients come in and waste gets out, has to go across the surface. So we're worried about a volume to surface area ratio. And as things get bigger and bigger and bigger, the volume increases faster than the surface area. So that dumb movie with this huge worm going through the sand or whatever it was, that can never happen because worms breathe through their skin. It would have a bad surface to volume ratio. Okay, small cells have a greater ratio of surface area, so you can have small worms just fine. You can see here, you can see the amount of surface area, and this will be deep as well, on a small cell, right? So you have all this surface area in the middle if you have small cells. And if you just have one big cell, all you get is the outside surface area. That's not enough to move nutrients in and waste out. So you can never get the blob. You can't just get this giant cell. It wouldn't be able to get its nutrients in and its waste out efficiently. Prokaryotic cells are structurally simpler than eukaryotic cells, right? We've said that there's no nucleus. What you get is a nucleoid region. You don't get a defined nucleus. You get a region. A eukaryotic cell has an actual nucleus with a membrane that's porous. You have organelles in here. So the eukaryotic cell is I don't want to say more advanced, but it's definitely more complicated. Prokaryotic cells are small and relatively simple. So, like we said, no nucleus, but they do get the flagella, remember, and this helps them move. They have a cell wall. Remember, they also can have that capsule that will help protect them. They also have the cell membrane, and then they don't have to have the pili or the flagella, but many of them do. So they have locomotion and they can stick to surfaces. Eukaryotic cells are partitioned into functional compartments. What that means is things are divided out so that they're more efficient. And so our big forms of life, all the things you can kind of see and think of, were eukaryotic cells, whether it's a plant or an animal. We have a real nucleus, and we have many more organelles. The prokaryotic cells are pretty much always bacteria. They're small. You really can't see them. So all the life you see is going to be eukaryotic cells. Okay, membranes form the boundaries of many of eukaryotic cells. Remember, that evolved probably from the membranes folding in. And then this compartmentalizing the interior of the cell facilitates the metabolic activities. It makes them much more efficient. It's not a blob. It's not all doing the same thing. You can have proteins being synthesized in one area. You can have vacuoles holding things. It's a much more efficient form of life. Okay, you have a typical animal cell here. And they kind of just throw everything on this. So you have your nucleus, and it's a porous nucleus. This is where the genetic material is stored. This is the brain of the cell. You have your rough endoplasmic reticulum, and remember, that's going to have ribosomes in it, so that's what makes it rough. You're going to have your Golgi apparatus here. That's going to process your proteins and stuff and move them along. You have your mitochondria here. That's your energy. You have microtubules here, and that keeps everything where it's supposed to be. It's like a skeleton. Okay? A typical plant cell has some structures that animal cells lack. All right, so they have this huge central vacuole, and they use that to store water and keep the plant cell rigid. All right, obviously they have chloroplast. We do not photosynthesize. I could put you in the sun all day long and you're never going to make any sugar. They also have a cell wall. Now this is outside the cell membrane. We have a cell membrane. They get a cell wall that the cell kind of pushes up against to, and that gives them structure. We have a skeleton. They get a cell wall. All right, the nucleus is the cell's genetic control center. It's the brain. It's where all the DNA is that controls everything, and it's separated from the cytoplasm by a nuclear envelope. And that's a big deal because it protects the DNA from things that would degrade it. Out in the big bad world of the cytoplasm, there are actually enzymes that go around degrading nucleic acids, and that DNA can send messenger RNA out, and maybe the messenger RNA gets degraded real quick. Who cares? You have the main copy of the DNA protected.